now that we differentiated between predictive modeling, generative modeling, machine learning, and traditional statistics, we can dive a little bit deeper into terms within the field of machine learning. You see very frequently in this course and any learning materials, people talking about machine learning algorithm and a machine learning model. So those are two different things, right? So when you are implementing or developing a machine learning model, first you need a recipe, a set of steps, an algorithm to provide instructions to build your model, right? For example, linear regression is a methodology, is an algorithm to create a machine learning model, to create a linear regression model. There is also L1 regularized logistic regression as this example here. There is also principal component analysis. That's another method that will make your model fit to the data. There is random forest. That's another algorithm. So the methodology to create your model is the machine learning algorithm. And you will be learning about these machine learning algorithms throughout this course as well. You learn about different types of algorithms, how they work, what are the types of data and problems where they excel, and when they should not be used. Right? So hopefully this course will help you to develop this critical thinking about what type of machine learning strategy, technique, or algorithm will be the best for your data, for solving your problem, for answering your question. So just to give an example here, for an example of a typical linear or logistic regression algorithm, you're going to start with a spreadsheet of data where you have features on the columns, samples on the rows. This example here is just an illustration of a gut microbiome composition data. And the algorithm will start setting random parameters in your model, or weights, and random bias as well. I'm going to dive a little bit deeper into what those terms mean soon. And it will use a function that will tell how wrong the model is. And this algorithm will learn those parameter values from the data. We use the data to learn these parameter values to derive the most accurate model possible. And that model resulting from this algorithm will be doing the predictions. Well, the model is the what has been learned from this machine learning algorithm. Summarizing, you can define the algorithm as a set of rules or a recipe, a series of steps that the computer will follow to achieve a specific goal. In this case, it's creating a machine learning model. And then the model is a specific mathematical function learned from the data that maps these inputs or predictors into predicted outcomes, or clusters if you're doing clustering analysis. So once your algorithm trains the model, derives this model, then you end up with a mathematical formula like that, which is basically your model. It can, for example, learn in this hypothetical example that these floating points here, floating point numbers multiplied by these taxa abundances will give a probability of a patient or a participant of a case control study cohort will be a case or a control, right? So that's what the algorithm achieved. It created this mathematical model, this machine learning model that can now make predictions for you. And just to illustrate a little bit of how the input data would look like if you want to provide this input data to make the machine learning algorithm teach the model how to make accurate predictions. You would pretty much see a contingency table or a matrix like that, a two-dimensional rows by columns, a spreadsheet. If you're familiar with Excel, for example, you would have a series of rows where each row here would be a different sample or a different individual in a study cohort. So for example, S01 is individual 1, S02 is individual 2, and then you have as many as the N or the size, sample size you have in your study. And then in the columns, you would have a different predictor. In the case of gut microbiome example that I'm using here, you would have like different genus or species in each column. In case of transcriptomics, you would have different genes. In case of metabolomics, you would have different metabolites in the columns. Okay? And then in the cell of each one of these combinations of rows and columns, you would have the values. Here in the gut microbiome example, these values are the relative abundance, the percentage of that microbe in the gut. So for example, Bacteroides for sample 01 has 18, represents 18%, 18.4% of the gut microbiome total composition. So if you are just trying to understand how these samples group together, you would not need outcome information if you're just trying to do clustering, unsupervised learning that you'll learn more about later. But if you want to make predictions of certain outcomes that are associated to those predictors, you would have one additional column in your data, which would be the outcome, which could be case control, for example, case if the person here, the sample one has a disease, control if it doesn't, or it could be a different type of outcome, like in human genomics, you can even predict BMI from genetics. You can even predict age from epigenetic data. For example, you can use DNA methylation as an indication of age. So there are different continuous or categorical outcomes that you can leverage for predictive like classification or regression tasks. Or if you don't have that outcome, you can use unsupervised learning like clustering, PCA, principal component analysis and clustering, to try to group these samples by the patterns that they share and try to identify patterns that they share that could be an indication of certain outcomes of interest that then you would decide to further look into it or further collect more data. And just some key additional terminology here. When we are talking about the predictors or the columns in your data set, in machine learning, we call those features, which is basically the input or the specific informative variables or predictors used for your model training and for inference for prediction later. They're also known as the independent variables. And when we are talking about those outcomes, we usually call them target or output, which are the specific outcome the machine learning model is trying to predict, also known as the dependent variable. So just to summarize here, the big picture, the overview of how a machine learning study workflow would look like. You would start from your data first. You would prepare your data, put in that specific format that would be machine learning ready. You may want to do some normalization, some quality control, of course, so you don't generate a biased model because of biased data. So you start understanding and preparing your data. Then on the next step, you decide which algorithm to use, the machine learning algorithm that will create your model, and then you run this algorithm to derive a model, to derive this mathematical function that will make the predictions or identify patterns, make new hypotheses about your data for you. So once you have a good model, let's say you have good data, you train and test your model is working really well. So now why do I want this model? What can I use it for? Once you have a good model, it's time to test and deploy your model, which means like model deployment means pretty much means to take your model and perform inference on new data, on unseen data. So you could, for example, if it's a clinical microbiology model that's trying to predict the risk for future disease in patients that have not been diagnosed yet, you could collect new samples from these patients, extract the same sort of input data, but instead of training the model with this data, you use that pre-trained, that already trained model that you developed to make a new prediction. So that's the inference portion of your model. In this stage, the model is not learning anymore. It's leveraging what it learned to make new predictions, and it will tell you the probability of that patient to have the disease under certain conditions at a certain age, for example. And if this patient has a high risk according to your model, then you could, for example, do a more careful follow-up with this patient, or do preventive care depending on the type of disease. So, for example, I worked a lot on coronary artery disease. If we see that the patient has a high risk of having a heart attack before the 40s because of high cholesterol or high artery calcification, then the preventive care would be to administer a certain dose of statin to the patient. So then he could postpone that outcome, or even never have that outcome of high risk. That's just an example of how the model would be useful after you develop that model.